History doesn't just disappear. It leaves evidence, clues, whispers from the past that most people walk right by. But not us. I'm a time detective, and these ancient mysteries are my cold cases. Some have remained unsolved for thousands of years until now. Welcome to Adventures Past, where we don't just study history, we crack it wide open. In 79 CE, Mount Vesuvius entombed an entire city in volcanic ash. For nearly 2,000 years, we thought we knew their stories. A mother clutching her child, two sisters in a final embrace, tales of family love frozen in time. But in 2024, scientists extracted DNA from teeth preserved in volcanic ash. What they found shattered every assumption we had about these ancient victims. The mother? Not a mother. Not even a woman. The child? A stranger from another land. Everything we thought we knew was wrong. Today's investigation takes us into the microscopic world of ancient DNA, genetic fingerprints left at history's greatest crime scenes. Armed with cutting-edge technology that would seem like magic to our ancestors, we're about to expose the truth behind 5 of history's most misunderstood mysteries. From the ash-covered streets of Pompeii to Viking burial grounds, from plague-riddled medieval towns to Egyptian tombs, we're following a trail of genetic evidence that rewrites everything you learned in history class. Fair warning, what you're about to discover might be disturbing. Not because it's gruesome, though some of it is, but because it reveals how wrong we've been. How our assumptions, biases, and romanticized stories have hidden the truth for centuries. So grab your magnifying glass and prepare to question everything. Our first stop? The most famous archaeological site in the world, where DNA evidence has just blown the case wide open. August 24th, 79 CE, for the residents of Pompeii, it started as an ordinary morning. Within 24 hours, the entire city would be buried under 20 feet of volcanic ash and pumice. But the pyroclastic flows that killed these people also performed an act of accidental preservation that would puzzle investigators for 2 millennia. In 1863, archaeologist Giuseppe Fiorelli made a groundbreaking discovery. The bodies had decomposed, leaving perfect cavities in the hardened ash. By pouring plaster into these voids, he created detailed casts of Pompeii's victims in their final moments. For over 150 years, these ghostly figures told stories that touched millions of visitors. Stories of love, family, and human tragedy. Stories that, we've just discovered, were almost entirely fiction. Enter our modern investigative team. November 2024, a joint task force from Harvard Medical School, the University of Florence, and the Max Planck Institute announces results that would shake the archaeological world. They had successfully extracted and analyzed DNA from 14 of the 86 plaster casts undergoing restoration. The key to their breakthrough? Teeth. While flesh and bone had long decomposed, dental pulp protected inside tooth enamel had survived nearly 2,000 years. Like a genetic time capsule, waiting to reveal its secrets. Let's examine our first case, the House of the Golden Bracelet. For decades, tour guides have pointed to one of history's most heartbreaking scenes. An adult wearing an elegant golden bracelet, cradling a small child. The narrative was irresistible. A wealthy mother, perhaps, trying to protect her child in their final moments. But when geneticists examined the DNA, the story crumbled like ancient papyrus exposed to air. The adult with the golden bracelet? Genetically male. The child? No biological relation whatsoever. In fact, none of the 4 individuals found together in that house were related by blood. Think about that for a moment. Every museum placard, every textbook, every documentary that described this mother and child, all based on nothing more than assumptions. We saw a bracelet and assumed woman. We saw an adult holding a child and assumed a parent. The DNA tells us these were simply 4 unrelated people who happened to die in the same place. But the revelations were just beginning. In another house, the house of the Cryptoporticus, lay two figures locked in what appeared to be a tender embrace. For over a century, they've been known as the Two Sisters, or sometimes the Two Maidens. Visitors would pause, moved by this display of familial love in the face of death. The DNA results? At least one of them was male. No maternal lineage connecting them. No evidence they were even from the same region. Just two young people who found each other in their final, terrifying moments. What makes these discoveries even more intriguing is what they reveal about Pompeii itself. The genetic analysis showed something historians had suspected but couldn't prove. Pompeii was remarkably cosmopolitan. Most residents descended from immigrants who had arrived from the eastern Mediterranean. Despite centuries of conquests, invasions, and political upheaval, the genetic continuity was striking. The man with the golden bracelet? His DNA showed ancestry from the eastern Mediterranean with genetic markers suggesting North African heritage. He had black hair and dark skin, a far cry from the pale Roman matron tour guides had described for decades. This wasn't just about correcting a few mislabeled museum displays. This was about understanding the very nature of Roman society. Pompeii wasn't a homogeneous Italian town. It was a melting pot, a crossroads of cultures, more like modern New York than the ethnically uniform city we'd imagined. The technical achievement here deserves its own recognition. Extracting readable DNA from bodies subjected to temperatures exceeding 500 degrees Celsius should have been impossible. Yet through painstaking work, using techniques that didn't exist even 5 years ago, scientists reconstructed entire genomes from microscopic fragments. They used advanced methods to identify and isolate endogenous DNA, that is, DNA actually from the ancient individuals rather than modern contamination. Through specialized laboratories with clean rooms more sterile than operating theaters, they pieced together genetic puzzles with fragments, sometimes only 30 base pairs long. But perhaps the most profound revelation is this. Of the 14 individuals tested so far, the majority showed no family relationships despite being found together. This challenges our fundamental assumptions about how people behave in disasters. We imagine families huddling together, but the DNA suggests many died among strangers. What does this mean? Perhaps in those final chaotic hours, social bonds mattered more than blood ties. Perhaps servants died alongside masters, friends alongside neighbors, strangers who became companions only in their final moments. The DNA can't tell us their relationships, only that they weren't genetic. The human stories of friendship, employment, or mere proximity remain locked in the ash. There's something unsettling about these revelations. Every time we look at these casts now, we must question what we see. Is that protective gesture really maternal love? Or something else entirely? Are those intertwined figures lovers, friends, or strangers, seeking any human contact in their terror? The investigation continues. Of Pompeii's 104 casts, only 14 have been analyzed. Each extraction takes months of careful work. Each result potentially overturns another cherished story. As we speak, scientists are selecting the next subjects for analysis, knowing that each test might shatter another 2,000-year-old myth. But Pompeii's DNA mysteries, shocking as they are, represent just one piece of our investigation. Because halfway around the world, in the frozen grounds of Scandinavia, another genetic revolution was about to destroy everything we thought we knew about the Vikings. Close your eyes and picture a Viking. I'll wait. Let me guess. Tall, muscular, long blonde hair, piercing blue eyes, probably wearing a horned helmet. Now open your eyes, because I'm about to show you how Hollywood, historians, and our own imaginations got it completely wrong. The year is 2020. Professor Eske Willerslev of Cambridge and Copenhagen universities releases the results of the largest Viking DNA study ever conducted. 6 years of work, 442 Viking skeletons from over 80 sites, Samples spanning from 750 to 1050 CE, covering everywhere from Greenland to Ukraine. What they found sent shockwaves through conference halls from Stockholm to Dublin. The Vikings, those legendary blonde warriors of the north, most of them had dark hair and brown eyes. But that was just the beginning of the genetic plot twist. Let's visit our first crime scene, a Viking burial ground in Orkney, Scotland. The bodies were found with all the traditional markers of Viking warriors. Swords with distinctive Nordic designs, shield bosses, boat-shaped graves oriented toward the sea. For decades, archaeologists confidently labeled this a Viking raiding party cemetery. The DNA results. These Vikings were 100% Scottish Picts, not a drop of Scandinavian blood. They were locals who had adopted Viking culture, burial practices, and probably fighting techniques. They lived as Vikings, died as Vikings, were buried as Vikings. But genetically, they were as Scottish as haggis. Think about what this means. Viking wasn't an ethnicity. It was a job description. It was a lifestyle. It was a brand. And like any successful brand, it attracted franchisees from across Europe. The genetic diversity uncovered by this study reads like a casting call for a very different Viking series. Southern Europeans with Mediterranean complexions, Western Asians with genetic roots in the Caucasus, indigenous Sami people from the Arctic, all of them adopting the Viking way of life, joining crews, raiding coastlines, and eventually being buried with honors as Norse warriors. But the most heartbreaking discovery came from Estonia. 2 boats buried in the sand around 750 CE, 41 Swedish males, all killed in what appeared to be a single violent event. The archaeological evidence suggested a Viking raid gone catastrophically wrong. When scientists analyzed the DNA, they found something that transformed these ancient statistics into a human tragedy. 4 of the men were brothers, multiple other sets of close relatives. Isotope analysis of their teeth showed they all grew up in the same small region of Sweden, possibly even the same village. Picture that for a moment. A small Swedish community sends its men on what should have been a profitable raid. Brothers fighting side by side, cousins watching each other's backs, friends who'd grown up together, now seeking fortune in foreign lands. None of them would return home. The DNA revealed they died the same day, probably within hours of each other. The careful arrangement of bodies in the boats suggests the survivors took time to honor their dead before fleeing. These weren't anonymous raiders. They were someone's sons, brothers, husbands. An entire generation of a small community erased in a single day. The migration patterns revealed by the study read like ancient flight records. Norwegian Vikings predominantly went west to Ireland, Scotland, Iceland, and eventually Greenland. Their DNA still runs strong in these regions. In fact, 6% of the UK population today carries Viking DNA markers, with hotspots in the Scottish Islands reaching up to 25%. Danish Vikings focused their attention on England. The genetic evidence perfectly matches historical records of the Danelaw, that portion of England under Viking control. Modern residents of York, Leicester, and other Danelaw cities show significantly higher Scandinavian ancestry than the rest of England. Swedish Vikings headed east, into the Baltic, Russia, and beyond. They established the trade routes that would become the foundation of Russian cities. Their DNA trail leads all the way to Constantinople, where Vikings served as the emperor's elite guard. But here's where it gets really interesting. Despite all this movement, despite Vikings from different regions meeting in trading posts and raid staging areas, they rarely intermarried. Norwegian Vikings in Ireland didn't marry Danish Vikings from England. Swedish Vikings in Russia maintained distinct bloodlines from their Western cousins. The Vikings were far more tribal than we imagined. Regional identities mattered more than a shared Viking culture. They might raid together, trade together, even die together. but they maintain distinct genetic populations that scientists can still identify today. One discovery stands out for its modern implications. Remember learning about the CCR5 gene mutation, the one that provides resistance to HIV? This mutation, carried by about 10% of modern Europeans, has been traced back through Viking bloodlines. It originated somewhere near the Black Sea around 700 BCE, but it was Viking migrations that spread it across Europe. A mutation that would save lives during the AIDS epidemic, distributed by warriors 1,000 years earlier. History's unexpected gift to the future. The study also demolished our image of Viking physical appearance. Genetic reconstruction shows a population with predominantly brown hair, ranging from light brown to black. Blue eyes existed, but were far from universal. Many Vikings had brown or hazel eyes. Their skin tones ranged from fair to olive to quite dark, especially among those with Southern European or Asian ancestry. The stereotypical blonde Viking? They existed, sure, but they were the minority. Modern Scandinavians are actually significantly fairer than their Viking ancestors. The genetic shift toward lighter features happened after the Viking Age, not before. What caused our collective false memory of blonde Vikings? Partly, it's 19th century nationalism. As Scandinavian countries built their modern identities, they reimagined Vikings as pure Nordic archetypes. Nazi propaganda later amplified this false image, using Vikings as symbols of supposed Aryan supremacy. The real Vikings would have laughed at such ideas. They were opportunists who valued successful raiders regardless of their ancestry. A Pictish warrior who could swing an axe was more valuable than a blonde Swede who couldn't. Merit mattered more than melanin. The genetic evidence also reveals Vikings as history's entrepreneurs. They established trading networks that spanned continents. They adopted useful technologies from every culture they encountered. They were multilingual, multicultural, and magnificently adaptable. One fascinating detail: Viking women traveled far more than previously thought. While male DNA dominates in raiding parties, settlement sites show substantial female migration. Viking women established homes in Iceland, Greenland, and the Scottish Islands. They brought Scandinavian culture, language, and yes, genes to these new frontiers. But not all Viking stories have been decoded yet. Ongoing studies are examining Viking settlements in North America, looking for genetic traces in indigenous populations. Others are studying the mysterious disappearance of Greenland Vikings, seeking genetic clues to their fate. Each study reinforces the same truth: the Vikings were never a pure ethnic group spreading Nordic genes across Europe. They were a diverse confederation of warriors, traders, and settlers, united by ships and ambition rather than blood. As we close this chapter of our investigation, consider how this changes everything. Every Viking movie, every museum display, every historical novel, most are depicting a blonde, blue-eyed fantasy that never existed. The real Vikings were far more interesting, a multicultural force that shaped Europe not through genetic purity, but through cultural dynamism. But if Viking DNA surprised us, what we're about to uncover in the plague pits of Europe will leave you questioning the very nature of human survival. Because locked in the teeth of plague victims, scientists have found evidence of an evolutionary arms race that shaped who lived, who died, and why you're alive today. Some crime scenes aren't marked by violence, but by silence. Empty villages, mass graves, entire civilizations that simply stopped. For centuries, we could only guess at what invisible killer struck them down. Now, thanks to DNA extracted from ancient teeth, we're finally identifying history's most prolific serial killer. Our investigation begins in 2024 in a state-of-the-art laboratory where scientists made a chilling discovery. They were analyzing the Black Death bacterium, Yersinia pestis, when they noticed something odd. The plague that killed 1/3 of Europe in the 14th century had evolved a specific gene mutation that made it less deadly over time. Wait, less deadly? That seems backwards. Wouldn't evolution favor a more lethal pathogen? Here's where our detective story takes a sinister turn. The mutation in the PLA gene didn't make the plague weaker, it made it smarter. By keeping victims alive longer, the disease could spread further. A victim who dies in 2 days infects their household. A victim who lingers for a week infects their entire neighborhood. The plague had learned the first rule of successful parasitism. Don't kill your host too quickly. But this 14th century pandemic wasn't plague's first rodeo. Let's rewind to 541 CE. The Byzantine Empire under Justinian was at its height. Constantinople was the jewel of the civilized world. Then people started dying. Historians called it the Plague of Justinian, and it killed an estimated 25 to 50 million people, a quarter of the world's population. For 1,500 years, we didn't know if this ancient plague was the same disease that would later cause the Black Death. Then, in 2014, scientists extracted DNA from the tooth of a plague victim buried in Bavaria. The genetic fingerprint was unmistakable. Yersinia pestis. Same killer, different century. But the DNA revealed something unexpected. This wasn't an African disease, as historians had long believed. The genetic trail led back to Central Asia. The plague had traveled the Silk Road, hitching rides with merchants and their black rat stowaways, reaching Europe just as the Byzantine Empire peaked. The timing wasn't coincidental. Increased trade meant increased disease transmission. Prosperity became vulnerability. The very connections that made Constantinople rich also made it a perfect plague incubator. But here's where our investigation takes an even darker turn. In 2024, Swedish scientists analyzing 5,000-year-old skeletons made a discovery that rewrote prehistory. They were investigating the mysterious collapse of Neolithic Europe, when the continent, as first farming civilizations vanished around 3000 BCE, history's first genocide, climate catastrophe. The DNA told a different story. In 17% of tested remains from Sweden and Denmark, they found Yersinia pestis, the plague. But this wasn't the medieval version. This was Plague 1.0, a prototype that would eventually evolve into history's greatest killer. This Stone Age plague lacked the flea transmission gene that would make later versions so deadly. It spread differently, probably through direct contact or contaminated food. But it was still lethal enough to potentially trigger the collapse of Europe's first agricultural societies. Think about that. 5,000 years ago, before the pyramids, before writing, before bronze tools, the plague was already shaping human history. Entire cultures vanished, leaving only mass graves and abandoned settlements. For 50 centuries, we didn't know why. Now, thanks to DNA, we've identified the killer. The investigation revealed 3 distinct infection events across prehistoric Scandinavia. This wasn't a single outbreak, but waves of disease, each potentially wiping out entire communities. The farmers who had brought agriculture to Europe faced an enemy they couldn't see, couldn't understand, couldn't fight. But the most shocking discovery came from Egypt. For decades, scientists assumed plague was a Eurasian disease. Africa, separated by the Sahara, should have been protected. Then, in 2024, researchers analyzing a 3,290-year-old Egyptian mummy found something that shouldn't have been there. Yersinia pestis DNA. This wasn't just any mummy. The individual, an adult male, showed advanced stage plague infection in his intestinal tissue. He'd been sick for days, maybe weeks. The infection had time to spread throughout his body before death. The date is significant. 1290 BCE. This was the height of Egypt's new kingdom. Ramesses II had recently died. Egypt was still a superpower. Yet plague had already crossed the desert, establishing itself along the Nile 3,000 years before the Black Death. Ancient Egyptian medical papyri suddenly make more sense. Descriptions of painful swellings in the groin and armpits, classic plague buboes, accounts of sudden fevers that killed within days, prayers to protect against the disease that comes in the darkness. They were describing plague, but until now, we didn't recognize it. The DNA evidence suggests plague reached Egypt via trade routes, probably through the Sinai. The same merchant ships that brought cedar from Lebanon and copper from Cyprus also brought death. Egypt's ports, bustling with international commerce, became disease entry points. But here's what makes this discovery truly significant. If plague reached Egypt 3,300 years ago, where else did it spread that we haven't discovered yet? How many mysterious population collapses? How many abandoned cities? How many lost civilizations were actually plague victims? The genetic analysis revealed another disturbing pattern. Each plague outbreak left its mark on human DNA. Survivors weren't random. They were people with specific genetic variations that provided resistance. Over millennia, plague shaped human evolution, selecting for genes that offered protection. One example, the CCR5 delta 3-2 mutation we mentioned with Vikings. This mutation, which provides HIV resistance today, may have originally evolved as plague protection. Communities hit hardest by plague show higher frequencies of this mutation in modern populations. The Black Death literally changed our genetic code, but plague wasn't done surprising us. In 2025, analysis of medieval plague victims revealed that the bacterium was actively evolving during the pandemic. Different waves of the Black Death were caused by slightly different strains. The plague was mutating in real time, adapting to human defenses, finding new ways to spread. Some strains were more lethal, killing quickly. Others were better at transmission, keeping victims mobile longer. The pandemic wasn't a single event, but an evolutionary battlefield, with the plague constantly probing for weaknesses in human biology. The investigation also solved a long-standing mystery. Why did plague disappear from Europe after the 18th century? The DNA suggests it didn't disappear, it evolved. Modern plague strains are less transmissible in European climates. The bacterium adapted to new environments, new hosts, new transmission routes. It's still out there, still evolving, still waiting. What's truly terrifying is how DNA analysis revealed plague's presence in unexpected places. Neolithic settlements thought destroyed by warfare? Plague victims. Bronze Age trade centers that mysteriously emptied? Plague. Iron Age Hilforts abandoned overnight? Often plague. Each discovery reinforces the same truth. Infectious disease has shaped human history more than any war, any emperor, any ideology. Civilizations that seemed invincible fell to an enemy measured in microns. Trade routes that brought prosperity also brought death. Progress and plague traveled hand in hand. The DNA evidence also revealed plague reservoirs, wildlife populations, where the disease persisted between human outbreaks. Central Asian marmots, African gerbils, North American prairie dogs. These animals harbored plague for centuries, occasionally spilling over into human populations with catastrophic results. Understanding these reservoirs helps explain plague's seemingly random appearances. A change in climate drives rodents toward human settlements. A new trade route connects a plague reservoir to virgin populations. Suddenly, a disease dormant for decades explodes across continents. Modern plague still kills about 600 people annually. It's treatable with antibiotics, but only if caught early. In regions without modern healthcare, mortality rates still approach medieval levels. The killer that shaped human history isn't gone. It's just constrained by modern medicine. But perhaps the most profound revelation is how plague DNA reveals human resilience. Every outbreak left survivors. Every pandemic selected for resistance. We are the descendants of those who faced history's deadliest disease and lived. Our DNA carries the scars of ancient plagues and the keys to surviving future ones. As we close this chapter, Consider how many of history's mysteries might actually be disease events. How many lost civilizations fell not to barbarians, but to bacteria? How many demographic shifts, cultural changes, and historical turning points were actually shaped by invisible killers? The answers lie waiting in ancient DNA, in teeth and bones of victims who died centuries before anyone understood germs. Each extraction reveals new chapters in the world's longest murder mystery, the story of how microscopic killers shaped macroscopic history. But if plague DNA revolutionized our understanding of disease, what we're about to discover in Egyptian tombs will challenge everything you think you know about ancient populations, family relationships, and the very appearance of history's most famous civilization. The year is 1907. Two mummies arrive at the Manchester Museum. Labeled as brothers from 1800 BCE, their coffins bore inscriptions claiming they shared the same mother and father, Kunum Nakt and Nakdank, sons of a local governor. For over a century, they were displayed as brothers, their story touching millions of museum visitors. But Egyptologists noticed something odd. The two mummies looked nothing alike. Different skull shapes, different skeletal structures. Were they adopted? Stepbrothers? Or was something else going on? The mystery lingered for 111 years. Then, in 2018, scientists at the University of Manchester decided to extract DNA from the mummy's teeth. What they found would spark a family scandal 4,000 years in the making. The mitochondrial DNA, inherited only from mothers, was identical. These men shared the same mother. But the Y chromosome DNA, passed from father to son, told a different story. Kanumnakt and Nakdank had different fathers. The brothers were half-brothers. Their mother had children with two different men. The ancient inscriptions calling them sons of the same father? A 4,000-year-old lie, preserved in hieroglyphs. Why the deception? In ancient Egypt, inheritance passed through the father's line. By claiming the same father, both brothers could inherit equal status and wealth. Their mother had orchestrated a fraud that would last 4 millennia, only exposed when modern science peered into their DNA. This discovery opened floodgates of questions. If these brothers lied about their parentage, how many other mummy relationships are fiction? How many ancient families harbored secrets they thought would be buried forever? But the Manchester brothers were just the beginning. In 2017, a team of German scientists published results that would overturn everything we thought we knew about ancient Egyptian genetics. They had analyzed DNA from 151 mummies from Abusir el-Melik, a site south of Cairo, spanning 1,300 years of Egyptian history. What they expected to find was genetic continuity, ancient Egyptians looking pretty much like modern Egyptians. What they found instead sent shockwaves through the archaeological community. Ancient Egyptians were genetically closer to ancient Near Eastern and European populations than to modern Egyptians. They clustered with ancient populations from the Levant, Anatolia, and Europe. Sub-Saharan African ancestry, which comprises 15 to 20% of modern Egyptian DNA. In ancient Egyptians, it was almost non-existent. The implications were staggering. Those Hollywood movies showing ancient Egyptians as they appear today? Genetically inaccurate. The ancient Egyptians who built the pyramids looked more like their Mediterranean neighbors than their modern descendants. But when did this genetic shift happen? The DNA timeline provided the answer: after the Roman period. For over 3,000 years, from the Old Kingdom through the Ptolemaic dynasty, Egyptian genetics remained remarkably stable. Then, sometime after 400 CE, sub-Saharan African genes began flowing into Egypt in significant numbers. The timing corresponds with the decline of Roman power and the rise of trans-Saharan trade routes. As the Mediterranean world fragmented, Egypt's connections shifted south. The genetic map reveals what historical records sometimes miss: the movement of people, not just goods and ideas. This discovery triggered fierce debates. Some argued the Abusir el-Melik mummies weren't representative of all Egypt. Others pointed out that Upper Egypt, closer to Nubia, might show different patterns. The scientists responded by expanding their studies, and each new batch of results confirmed the pattern. Ancient Egyptians were genetically Mediterranean, but the most mind-blowing discovery was yet to come. In 2025, scientists analyzing 7,000-year-old mummies from the Yuan Mahagig site in Libya made a discovery that rewrote the textbook on human genetics. These mummies, naturally preserved in the Sahara Desert, belonged to a completely unknown human lineage. This wasn't just another population group. This was a ghost population, humans who diverged from other African groups 150,000 years ago and remained genetically isolated for tens of thousands of years. They were living fossils, carrying DNA from humanity's deep past. The analysis revealed something extraordinary. While most human populations show 2 to 3% Neanderthal DNA from ancient interbreeding, these Saharan people had almost none. They had separated from other humans before the major out-of-Africa migrations that led to Neanderthal encounters. For 50,000 years, while the rest of humanity was spreading across the globe, mixing with Neanderthals and Denisovans, developing new technologies and cultures, this population remained in the Green Sahara, genetically frozen in time. They were arguably the most pure Homo sapiens ever discovered. Their DNA told the story of the Green Sahara, that period from 12,000 to 5,000 years ago, when the Sahara was a lush landscape of lakes and grasslands. These people thrived in this environment, developing a unique culture adapted to their isolated paradise. Rock art from the period shows them herding cattle, hunting abundant game, swimming in lakes where today there's only sand. But as the climate shifted and the Sahara dried, these people faced a choice: migrate or die. The DNA suggests most chose death over leaving their homeland. A few integrated with incoming populations, leaving faint genetic traces in modern North Africans. But as a distinct population, they vanished with the green landscape that sustained them. The discovery raises profound questions. How many other ghost populations existed in humanity's past? How many unique genetic lineages vanished without a trace? Each ancient DNA extraction might reveal another lost branch of the human family tree. Egyptian mummy DNA also revealed surprising insights about disease and adaptation. Ancient Egyptians showed genetic adaptations to malaria, confirming the disease plagued the Nile Valley for millennia. They carried genes providing partial resistance to tuberculosis, another ancient Egyptian scourge. Their DNA bore witness to thousands of years of evolutionary pressure from infectious diseases. One particularly intriguing discovery involved skin pigmentation genes. Ancient Egyptians carried variants associated with lighter skin than most modern North Africans, but darker than Europeans. They occupied a genetic middle ground, adapted to the intense sun of the Nile Valley, while maintaining the ability to synthesize vitamin D hair and eye color genes told their own story. While not as diverse as Viking populations, ancient Egyptians weren't uniformly dark-haired and dark-eyed. Red hair, while rare, existed. Light eyes appeared occasionally, especially in the Delta region with its closer connections to the Mediterranean. The mummy DNA also solved some historical mysteries. The famously fair-skinned portrayal of Queen Nefertiti? Genetically plausible. The red-haired mummy of Ramesses II? Not dyed with henna as some claimed, but natural genetic variation. Ancient Egyptian art, often dismissed as stylized, might be more accurate than we thought. But perhaps the most significant revelation was about ancient Egyptian society itself. The genetic diversity within single cemeteries was remarkable. Rich and poor, native and foreign, all buried side by side. DNA revealed a cosmopolitan society where ancestry mattered less than cultural adoption. Foreign mercenaries who adopted Egyptian customs were buried as Egyptians. Their children, genetically Greek or Nubian or Libyan, became fully Egyptian in culture, if not in blood. The DNA paints a picture of an inclusive civilization that absorbed immigrants while maintaining cultural continuity. This pattern held for over 3 millennia. Through invasions by Hyksos, Nubians, Assyrians, Persians, Greeks, and Romans, the core Egyptian genetic profile remained stable. Conquerors were absorbed rather than replacing the population. Egyptian culture proved stronger than Egyptian genes. The ongoing DNA analysis continues to surprise. Each mummy extracted reveals new secrets. Royal mummies thought to be relatives prove unrelated. Commoners show royal genetic markers. Family trees carefully constructed from inscriptions crumble under genetic scrutiny. One recent project analyzing Ptolemaic mummies, the Greek dynasty that ruled Egypt after Alexander the Great, revealed they maintained remarkable genetic separation from the native population for centuries. Despite adopting Egyptian customs and claiming to be pharaohs, they rarely married outside their Macedonian Greek ancestry. Cleopatra, the last pharaoh, was likely more Greek than Egyptian genetically. As we process these revelations, consider how DNA evidence forces us to reimagine ancient Egypt, not as an isolated African kingdom, but as a Mediterranean crossroads, not as a racially pure civilization, but as a genetic melting pot, not as a static society, but as a dynamic culture that absorbed influences while maintaining its core identity. The ancient Egyptians themselves seem to understand this. Their art depicts people of various skin tones working together. Their texts speak of foreign lands, but rarely foreign races. They define themselves by culture, not color, a lesson modern society might learn from. But if Egyptian DNA surprised us with its diversity and connections, our final investigation takes us to the far edges of the ancient world, where mummies in Chinese deserts and frozen Siberian graves are revealing impossible journeys and forgotten peoples that challenge the very foundations of human history. Our final investigation takes us to the edges of the known ancient world, where DNA evidence is revealing impossible journeys and forgotten peoples. These aren't footnotes to history. They're entirely new chapters that nobody knew existed. Let's begin in the Tarim Basin, the massive desert in western China. For decades, archaeologists have been puzzled by mummies found here. Dating back 4,000 years, these bodies were naturally preserved by the arid climate. But there was something unsettling about them. They looked European. Blonde and red hair, angular features, tall statures. Some wore tartans remarkably similar to Celtic designs. They were buried with cheese, wheat, and other Western foods. In the heart of ancient China, thousands of miles from Europe, lay people who looked like they'd stepped out of ancient Scotland. The mystery sparked international incidents. Chinese authorities, sensitive about claims to Western regions, restricted access. Western researchers pushed theories about ancient European migrations. New Age mystics claimed these were lost Aryans or Atlanteans. Everyone had a theory. Nobody had proof. Until 2021, when DNA analysis finally cracked the case. And the answer was stranger than any theory. These weren't Europeans. They weren't migrants at all. They were a local population that had lived in the Tarim Basin since the Ice Age. Their DNA revealed them as direct descendants of ancient North Eurasians, a population that contributed genes to both Europeans and Native Americans, but then largely vanished. The Tarim mummies were genetic time capsules, a population that had remained isolated in their desert oasis for over 10,000 years. While their distant cousins were populating Europe and the Americas, they stayed put, maintaining genetic patterns from humanity's deep past. But here's what makes this truly mind-bending. Despite their genetic isolation, they weren't culturally isolated. They buried their dead with wheat from the Near East, millet from China, cheese-making technology from the steppes. They wore textiles showing influences from across Eurasia. They adopted innovations from every direction while maintaining their unique genetic signature. It's the opposite of what we usually find. Typically, cultural exchange goes hand in hand with genetic mixing. The Tarim people managed something remarkable. They participated in the Bronze Age, global economy while maintaining genetic boundaries. They were ancient globalists who married locally. The DNA also revealed why they looked European, despite not being European. Those ancient North Eurasian genes they carried? They're the same ones that gave Europeans light skin and eyes. Convergent evolution made distant populations look similar. The Tarim people and Celtic populations evolved similar appearances independently, like two artists painting similar pictures without seeing each other's work. But the Tarim Basin held more secrets. Later mummies from around 2,000 years ago showed completely different DNA. These were actual migrants from the steppes, from East Asia, from South Asia. The original Tarim population had vanished, replaced, or absorbed by incoming waves. Their 10,000-year isolation had ended, and with it, their unique genetic legacy. What happened to them? The DNA suggests they didn't die out dramatically. They intermarried with newcomers, their genes diluting with each generation until their distinct signature disappeared. Today's Uyghur population carries traces of their DNA, mixed with dozens of other ancestral streams. The Tarim people live on, but only as genetic echoes. Now let's turn to an even more profound mystery: the Neanderthal population crash. In 2025, scientists analyzing Neanderthal DNA made a discovery that might explain one of prehistory's greatest mysteries: why our closest relatives went extinct. The study focused on an unusual source of DNA, the semicircular canals of the inner ear. These tiny structures, protected deep within the skull, preserved genetic material in specimens where other tissues had failed. What they revealed was catastrophic. Around 110,000 years ago, Neanderthal genetic diversity crashed. The population bottlenecked so severely that all later Neanderthals descended from a tiny group, perhaps just a few hundred individuals. For the next 70,000 years, until their extinction, Neanderthals carried the genetic scars of this near-extinction event. Low genetic diversity made them vulnerable. Harmful mutations accumulated. Fertility problems increased. Disease resistance decreased. They were the walking dead, genetically speaking, a species marked for extinction tens of thousands of years before they actually disappeared. What caused the bottleneck? Climate change is the leading suspect. The Eemian interglacial period ended around 115,000 years ago, plunging Europe into renewed cold. Forests retreated. Prey animals migrated or died. Neanderthals, perfectly adapted to the previous environment, suddenly faced a world they weren't prepared for. But here's the twist: they survived. For 70,000 more years, these genetically compromised Neanderthals hung on. They developed new technologies. They created art. They interbred with modern humans. They refused to go gentle into that good night. The DNA reveals their desperate strategies. Later, Neanderthals show signs of increased inbreeding, probably because suitable mates were scarce. They also show increased hybridization with modern humans, genetic rescue through interbreeding. Every human today with Neanderthal DNA carries evidence of these last-ditch survival efforts. But perhaps the most intriguing discovery involves what scientists are calling hidden human lineages. As DNA technology improves, we're finding genetic signatures of completely unknown human populations. Ghost populations that left no fossils, no tools, no art, only DNA traces in modern genomes. One example emerged from studying Tibetan populations. In 2025, scientists discovered that Tibetans carry DNA from an unknown hominin species. This wasn't Neanderthal or Denisovan. This was something else entirely. A third player in the human evolution game, previously completely unknown. The ghost DNA appears linked to high-altitude adaptation. Tibetans' remarkable ability to thrive above 4,000 meters might come from this mysterious ancestor. Somewhere in the heights of ancient Asia lived a human species perfectly adapted to thin air. They're extinct now, but their genetic gift lives on in every Sherpa who summits Everest. Similar ghost populations are emerging worldwide. Aboriginal Australians carry DNA from at least one unknown hominin. African populations show traces of archaic humans who diverged from our lineage over 500,000 years ago. Each discovery adds new branches to the human family tree, branches we never knew existed. The technology revealing these ghosts is revolutionary. Environmental DNA, genetic material extracted from soil, can reveal which species lived in a location, even without fossils. Cave sediments in Denisova Cave revealed multiple human species occupying the site over millennia, most leaving no skeletal remains. One cave in the Altai Mountains yielded DNA from Neanderthals, Denisovans, and modern humans, all from dirt. The soil remembered what the fossil record forgot. Every archaeological site potentially contains genetic archives in its sediments, waiting to be read. Protein analysis adds another dimension. When DNA degrades beyond recovery, proteins can survive. Collagen from a 1.9-million-year-old rhinoceros tooth recently yielded readable sequences. If similar preservation exists for hominins, we might push back genetic analysis to the origins of our genus. These techniques are revolutionizing our understanding of human migrations. The peopling of the Americas, long thought to involve a single migration across the Bering Land Bridge, now shows multiple waves. Some genetic signatures suggest coastal migrations. Others point to connections with Australasia that shouldn't exist according to traditional models. One jaw-dropping discovery: some Amazon tribes carry DNA most closely related to indigenous Australians and Papua New Guineans. How did Australasian genes reach South America? The conventional Bering Strait route leads away from Australia, not toward it. Either there was a previously unknown Pacific crossing, or the first Americans were far more diverse than we imagined. The DNA evidence keeps mounting for what scientists call the Paleo-American population, the first inhabitants of the Americas who looked nothing like modern Native Americans. Skulls like Luzia in Brazil and Kennewick Man in Washington state always seemed anatomically different. Now DNA confirms they represent a distinct population largely replaced by later migrations. But here's where it gets really weird. Some of this Paleo-American DNA survives in isolated pockets. The Sarui people of the Amazon carry higher percentages than any other Native American group. Living in one of Earth's most remote regions, they preserve genes from America's first colonizers. Each DNA study reveals the same pattern. Human history is far more complex than we imagined. Populations we thought were isolated were connected. Groups we assumed were related were strangers. Migrations went in directions that make no geographical sense. The genetic map looks like it was drawn by a madman, because it was drawn by the mad, beautiful chaos of human wandering. Consider the implications. Every indigenous group might harbor DNA from vanished populations. Every archaeological site might contain genetic surprises. Every assumption about human migrations might be wrong. We're not just rewriting prehistory, we're discovering it contains chapters we never knew existed. The Green Sahara people were just the beginning. As DNA technology improves, we'll find more ghost populations, more impossible migrations, more evidence that human history is stranger than human imagination. What drives this complexity? Perhaps it's the fundamental human urge to explore, to cross the next mountain, sail beyond the horizon, marry the beautiful stranger from the neighboring tribe. Our genes tell the story of restless wanderers who refused to stay put, who mixed and merged and moved in patterns that defy easy explanation. Or perhaps it's simpler. Perhaps humans, like water, simply flow wherever we can. Through mountain passes and across ice bridges, following coastlines and river valleys, carrying our genes like messages in bottles, washing up on distant shores to sprout new populations. The ancient DNA revolution has just begun. Each year brings new techniques, new discoveries, new mysteries. Genes we couldn't read yesterday become legible tomorrow. Populations we never knew existed emerge from genetic shadows. History itself transforms from a fixed narrative to a living mystery, constantly revealing new clues. As our investigation draws to a close, let's take a moment to appreciate the magnitude of what we've discovered. From Pompeii's misidentified victims to Vikings, who weren't Scandinavian, from plague DNA rewriting disease history to Egyptian mummies revealing ancient migrations, from Chinese desert dwellers maintaining impossible isolation to ghost populations hiding in modern genomes. Each discovery shatters assumptions we've held for generations. But perhaps that's the real lesson. The past isn't what we thought it was. It's stranger, more complex, more interconnected than our textbooks taught. Every certainty becomes a question mark. Every simple story becomes a multifaceted mystery. Some mysteries take lifetimes to solve, but we've just accelerated the investigation. If you found new evidence in this historical case file, join our research team by subscribing and sharing your theories below. The past isn't as buried as we thought, and with each discovery, we get closer to the truth. I'll meet you at the next excavation. This is Adventures Past, where history never truly dies. The evidence we've uncovered today will ripple through archaeology departments, rewrite museum placards, and force historians to reconsider everything they thought they knew. Because DNA doesn't lie. It doesn't care about our assumptions, our biases, or our cherished stories. It simply tells the truth, a truth often stranger than any fiction we could imagine. From the ash-covered streets of Pompeii to the frozen graves of Scandinavia, from plague pits to pyramid tombs, from Chinese deserts to hidden Amazon tribes, we've followed a genetic trail that reveals humanity's true story. Not a simple tale of distinct peoples in distinct places, but a complex web of wandering, mixing, isolating, and reuniting that defies any attempt at clean categorization. The time detectives have presented their evidence. The case files remain open because each new DNA extraction might overturn everything we've just learned. That's the beauty and terror of scientific investigation. Today's solution might be tomorrow's mystery. Until our next investigation, remember, question everything, assume nothing, and always follow the evidence, no matter how deeply it's buried or how radically it challenges what you think you know. This is Adventures Past, signing off. The past is calling, and it has more secrets to share.